This is the BioCentury Show.
Brought to you by the 5th East-West Biopharma Summit in South Korea. In March, explore how to globalize Asia innovation and connect with key players across Asia's innovation arc, from India to China to Japan.
Hello, welcome to the BioCentury Show. I'm Selena Koch, an executive editor at BioCentury, and I'm just so delighted to be joined today by Stephen Paul, one of the most influential leaders in drug development in the neurosciences, especially in psychiatry. By way of introduction, his early career was at the National Institutes of Health, after which he had a 17-year tenure, I believe, at Eli Lilly, and senior leadership roles in R&D. After retiring from Lilly in 2010, he joined Third Rock Ventures, and he has helped build and guide numerous neuroscience-focused biotechs, with names some of you all I think will recognize: Sage Therapeutics, Voyager, Rapport, and of course, Karuna Therapeutics, which was acquired by Bristol Myers Squibb for $14 billion for a schizophrenia therapy. Among the many things Steve has his fingers in today, he is board chair at a company called Seaport Therapeutics that has helped, that company's got back together some of the Karuna group to kind of take their momentum in a new direction. Now, that's a highly abbreviated summary of a very distinguished career. That's because I want to save our time for what's coming next, which is a discussion of how psychiatric drug development is changing, or not, heading into 2026. It's no secret, psychiatry is one of the toughest arenas for drug developers, but Steve has seen some successes. So, Steve, when animal models are poorly predictive, when the human genetics revolution that's been so useful in other areas has not produced a wealth of high-value targets in psychiatry, how do you go about identifying a biological mechanism that's, compelling enough to spend years pursuing?
Great. Well, first off, Selena, thanks for having me on the podcast. I really appreciate the invitation. But to get directly to your question, discovering medicines, I should say, for neuropsychiatric disorders like depression, anxiety, schizophrenia has been difficult, has been challenging. There's little doubt about that. The need is great, though. The unmet medical need, these are some of the most common afflictions of mankind. If you think of a disorder like schizophrenia, it affects about 1% of the population. So roughly 3.5 million people in the U.S. have schizophrenia. It is a chronic, very disabling disorder. We can get into the details a little bit later when we talk about Karuna, but it's one of the most disabling disorders that a human being can get. Depression, major depressive disorder, very common. The prevalence is roughly, 6-8% of the population. So again, and as disability life years go, it is also right up there at the top of the list. And of course, one of the major causes of suicide. And most people don't, appreciate that there are more suicides in this country than there are homicides every year. And a great deal of those suicides are due to untreated, inadequately treated depression, anxiety disorders, so on and so forth. So you're, affecting really roughly about 20% or more of the population with one of these major neuropsychiatric disorders. The medicines for these disorders have come about in a rather curious way. Many of them have been discovered by accident. Drugs used to treat one disorder tried in another, and they seem to work on like treating psychotic symptoms. That's how chlorpromazine, Thorazine, the very first antipsychotic drug, was discovered. Similarly, the first antidepressant drugs, the monoamine oxidase inhibitors, tricyclic antidepressants, they were discovered the same way. They were discovered by inpatients when these drugs were used, usually for other purposes, and then they were discovered in the case of the antidepressants to elevate mood, and they then became useful for the treatment of depression. Now, what has occurred in a much more scientific vein is that once the original drugs have been discovered by this phenotypic screening, as I call it, in humans, We then figure out how they work mechanistically. What do they do in the brain to produce their effects? And using that information, we can design new and improved drugs. But for the most part, with some very few exceptions, our Sage postpartum depression drug perhaps being one of those, they're really discovered almost by serendipity and what I call astute empiricism. In other words, that just not completely by accident, but there's been a lot of luck, a lot of serendipity involved in the discovery of these drugs. And then we've iterated and improved upon them over time. You're absolutely right. The biology of psychiatric disorders, what I call disease biology, is fairly primitive right now. I've been working on that in my spare time for many, many years, since my days at NIMH. We've learned some things. For example, many of these diseases do have a genetic component to it. They're heritable, but the genetics is very complex, very polygenic, if you will. And it's been difficult to say, okay, well, this is the target you need to go after to treat this particular disease. So we use this other approach most commonly. And fortunately, we've been able to develop some new medicines, particularly over the last few years, that I think has rejuvenated the field, given us more hope that we can come up with new and improved drugs, because that first generation of drugs that I alluded to, while effective, and it would be very difficult to treat someone with a serious depression without the current genre of antidepressants. It would be very difficult to treat psychotic disorders like schizophrenia without antipsychotic drugs that we do have. Nonetheless, these drugs leave a lot to be desired. They don't work in everybody. Yeah, go ahead.
Yeah, let me just break in. So
Sure.
this discussion of opportunism and serendipity, of course, it will resonate with everybody because it's true everywhere, even if it's especially true in psychiatry. But I do wonder if that Karuna model, right, taking something that has human proof of concept in the sense of it's been tried in the clinic before, but it had a liability, and then figuring out a workaround for that liability. I wonder if that's like, do you see that as a generalizable, is that going to be the dominant paradigm for a while?
I don't know whether it'll be dominant, but I do believe it'll be generalizable, and I believe there's much left to do there. And maybe the Karuna story would be a good one just to kind of summarize, really. We, at Lilly back in the early 90s, we were trying to develop a medicine that would improve memory and cognition in patients who had Alzheimer's disease. And in those days, we knew that the neurotransmitter acetylcholine, which, by the way, was the very first neurotransmitter discovered way back in the 20s, that was deficient in the brains of people who developed Alzheimer's disease, among other types of neurotransmitters. And we knew that that transmitter was involved in memory and learning. And the idea was that if you could boost the level of that neurotransmitter in people who had Alzheimer's, particularly early Alzheimer's, you would improve their memory. So symptomatically, you treat them. And it turned out that there were a number of ways of doing that. One is you could block the enzyme that metabolizes acetylcholine. Those are the so-called cholinesterase inhibitors, and that would boost the level of acetylcholine, and they had some modest effects. At Lilly, our strategy was to bypass the need for acetylcholine because it was chronically going down, degenerating with the disease, and maybe stimulate these receptors for acetylcholine, one class of which were called muscarinic receptors. And so we were developing a muscarinic receptor agonist, a drug called xanomeline, which stimulates several of the known muscarinic receptors. There are 5, and it preferentially stimulates 2 of them. And we were developing this drug to primarily improve memory in people with Alzheimer's, and took it all the way to a very large phase 2 trial, 343 patients, placebo, 3 drug doses, to see does it improve memory in patients with Alzheimer's. And the bottom line is it does, modestly. It did improve memory in patients. But we noticed something really quite unexpected. In about 25 to 30 to 35% of these patients, they had what we called, in those days, the behavioral disturbances associated with Alzheimer's disease. They literally developed psychotic symptoms, delusions, hallucinations, agitation, etc. And remarkably, when we treated these patients with this drug xanomeline, Not only did it improve memory, but these behavioral symptoms, these psychotic symptoms, also were markedly reduced within a couple of weeks of treatment. We also noticed this was a 6-month trial, a blinded trial versus placebo, drug versus placebo. We also noted that the emergence of these symptoms occurred in the placebo arm over that 6-month period to a much greater extent than they did in the drug-treated arm. Said another way, this drug xanomeline seemed to acutely treat the psychotic symptoms of Alzheimer's disease, but also prevented the emergence. They had a preventative effect. And this is something we didn't anticipate in the study. In fact, we introduced the measure of these behavioral symptoms to make sure the drug wasn't making them worse, but in fact, they made them much better. And that was our first clue that we could treat psychotic symptoms without the traditional dopamine blocking effects of antipsychotic drugs, which you don't want to use in this patient population if you can avoid them. And so we were quite amazed with these results. We, within the next few years, decided, well, this is very interesting. I'll come back to why we stopped this program in a minute. But we thought, well, let's see if this worked in people that had schizophrenia, who predominantly had these psychotic symptoms, hallucinations, delusions, etc. And sure enough, we did a small study, but we saw some rather remarkable improvement in these symptoms in patients with schizophrenia. Now, unfortunately, because this drug xanomeline was stimulating muscarinic receptors in the brain, producing, we believe, the beneficial effects, was also stimulating muscarinic receptors in the GI system, for example, and that caused a lot of nausea and vomiting and GI side effects that frankly limited our ability to advance that particular molecule. But what we discovered, and this was published way back in 1997, is that for the very first time, we could reduce psychotic symptoms through this very novel mechanism that did not involve blocking dopamine receptors, which has been traditionally the mechanism by which all other antipsychotic drugs work. And these drugs, the antipsychotic drugs, we were developing one at Lilly, a very, still to this day, widely used, a drug called Zyprexa, olanzapine. They also have very undesirable side effects too. They cause weight gain, very significant weight gain in a lot of patients. They produce a Parkinsonian-like syndrome called extrapyramidal symptoms, which can lead to a permanent neurological disorder called tardive dyskinesia. So ideally, having a drug that had none of that, but produced behavioral effects that were really quite robust, was very desirable. And that was the origins, if you will, of the work that we did at Karuna.
So at Karuna, you paired this with a peripherally acting antagonist to dial out the liability, and now there is an approved drug called Cobenfy on the market for schizophrenia, which is the first new mechanism in ages, which is a fantastic success story. But I feel like if you're going to the Karuna example, we also have to note that even though it had success in phase 3 as a monotherapy, it then didn't quite as an add-on therapy. And that just brings me into this whole territory of how do you actually test these things in a clinic to see what you need to see?
Yeah.
What can be learned from that?
Okay, but before we get there, let's talk just a little bit about what Cobenfy, what xanomeline plus trospium. You're absolutely right. The way around the problem was to design a combination drug where we combine xanomeline, which gets into the brain very well, with a drug that blocked these receptors but didn't get into the brain, something we call a peripherally restricted anti-muscarinic. And when we did that, we could dramatically dial down the side effects, allowing us now to go back to the clinic, 30 years later, to actually see if Cobenfy, which is what it's called now, but we called it CAR-XT, Karuna Xanomeline Trospium, could affect and beneficially affect the symptoms of schizophrenia, the psychotic symptoms. And indeed, that's what we demonstrated at Karuna. We had 3 positive randomized placebo-controlled trials, which in psychiatry is difficult to do right in a row. We were able to do that. And of course, that gave us really a lot of conviction around this mechanism. Now, before I get to your specific question about the add-on trial, the adjunctive trial, let me also mention something I failed to mention that's really important. And that is when we think of the symptoms of schizophrenia, we do think of positive symptoms, psychotic symptoms, predominantly, because that's kind of what gets patients into hospital. They're hallucinating, they're delusional, they have paranoid ideation, they think that people are after to get them, the FBI, the CIA. It's very strange, very bizarre. Almost anybody who sees this will know these people have a serious problem. But it actually is the other two symptom domains. They have what's called negative symptoms. So when their psychotic symptoms have sort of died down, they have social withdrawal. They have they have almost anhedonia. They don't experience pleasure. They're just sort of like zombies, to be honest with you. Part of this may be due to the medicine, but also part of it is due to the disease. And then not well known by the public, but very well known by psychiatrists, they develop a cognitive impairment. They develop a kind of a dementia. In fact, the old-time psychiatrists, many, many years ago, called it dementia praecox, dementia of the young. And so their learning and memory are impaired, particularly working memory. Their executive functions are impaired dramatically. And that's why only about 10% of people with schizophrenia ever become gainfully employed. They're that impaired. Now, here's the other interesting thing about the muscarinic compounds like Cobenfy. They seem to treat these other symptom domains, not just the psychotic or positive symptoms, but also the negative and cognitive symptoms. And I think still to this day, that fact, which needs to be confirmed and with additional studies, has not been fully appreciated. And when that
Well, it hasn't been quite as strong as the effect on positive symptoms, right?
Well, no, it's pretty robust, but the way the FDA handles those symptoms is that you have to design a trial where you enroll patients who have very low positive symptoms, but who have what we call predominant negative or cognitive symptoms. But in our emergent trials at Karuna, when we built in instruments to measure negative symptoms and we built in instruments to measure cognitive symptoms, when we looked at patients who had lots of negative symptoms or lots of cognitive symptoms coming into the trial, there was a dramatic improvement in those symptom domains. And that's all been published in peer-reviewed literature. And personally, as an old shrink, an old psychiatrist, I think that's the most interesting factoid about Cobenfy that I think will eventually become very important to practicing psychiatrists in treating this syndrome we call schizophrenia. So that's a very important finding. Now, back to your question, however, or your observation, we were also very interested in seeing whether if we added Cobenfy to standards of care where patients were on another antipsychotic, a more traditional antipsychotic, but who were still symptomatic. These were what we call inadequate responders. If we added Cobenfy, which is a mechanistically unique drug, would we see improved symptom control with the addition of this mechanistically distinct compound? And in that study, the results were kind of frankly ambiguous. The overall results were negative, but if you looked at the data and you removed patients who were on risperidol, which is one of the more commonly used antidepressants, I think it was about 30 to 40% of this trial. But if you looked at the other 60 to 70%, there was a very nice enhanced improvement. These studies, these add-on studies, quite frankly, are very difficult to carry out because you have to have a very stable baseline of people on their traditional antipsychotic before they enroll. And sometimes that waxes and wanes and sometimes is not optimized. So I think, frankly, Selena, I think the verdict is still out on whether this drug will adjunctively improve schizophrenia. They're going to have to do some more work on that. Of course, the other big indication is dementia-related psychosis, where about really roughly 40 to 50% of people who have Alzheimer's disease eventually develop these behavioral symptoms that we saw way back in the early 90s in the original xanomeline trial. These symptoms are arguably among the most problematic. And these are the symptoms that often get patients into nursing homes, into a care setting where these symptoms can be dealt with, can be handled. So the verdict there is still out. Those studies are underway. Bristol has said that they will read out sometime, I think, late this year, if I'm not mistaken.
That's
And
right.
that will be a very important milestone in the history of this medicine, xanomeline/trospium. My feeling on it is it should work because we initially found these antipsychotic effects in this same patient population. But again, logistically, these are difficult trials to carry out. Now, there are hundreds of patients now, many, many sites. So you get a lot of noise, a lot of variability. But I have my fingers crossed because this would be the medicine you'd want to use to treat these patients, because the compounds that we have today, the medicines that we have today, which are the antipsychotic drugs I've mentioned before, these drugs are by and large not very good to use in this patient population. They increase morbidity, they increase mortality. Unfortunately, we don't have anything else to control these symptoms, and they become very problematic.
And that is a good note with which to take a pause, because we really do need to go to a break now, and we'll be back in just a few moments. So stick around. We're going to dive into other areas of psychiatry, including psychedelics, kind of a hot topic right now. And we'll be
Exactly.
right back.
The BioCentury Show is brought to you by the 5th East-West Biopharma Summit in South Korea. An arc of innovation is emerging across Asia, and Western biopharma leaders are taking note, from cross-border deals to new co's. In March 2026, the 5th BioCentury Bay Helix East-West Biopharma Summit visits South Korea for the first time. Meet the biopharma leaders putting Korea innovation on the global map. Learn why Korea has become a clinical trial and manufacturing hub. Discover if Korea is the next hotspot for new co-formation. Plus, meet biopharma innovators from India to Singapore to China and Japan. Register now at biocenturyeastwest.com.
Welcome back to the BioCentury Show, where we are talking with Steve Paul, an entrepreneur and leader in drug development for the neurosciences. And today we're discussing psychiatry. So when we left off, we were talking about the path, the journey of Karuna and its new mechanism for schizophrenia. I want to turn now to the other very big disorder in psychiatry, depression. Well, of the things that are kind of bubbling up right now, emerging, what do you find particularly compelling in the depression space?
Well, there's a lot happening in the treatment of depression right now, which is fortunate because this is such a disabling and unfortunately not uncommonly fatal disorder. And so just to name a few that come top of mind, the work that we did at Sage Therapeutics was arguably the very first rational drug discovery effort in psychiatry. We discovered that a metabolite of progesterone, a compound called allopregnanolone. This was work that I did at NIH like in the mid 80s. We discovered back then that this drug actually was very active at what we call GABA receptors. GABA receptors are receptors for the major inhibitory neurotransmitter in brain, which was quite surprising. This is a progesterone metabolite. Progesterone works by working on a nuclear receptor that is basically a transcription factor to increase gene expression. And what we observed in those days was that this metabolite had very strong activity in rapidly affecting the excitability of the brain through enhancing this inhibitory neurotransmitter GABA. And to make a long story short, other work had postulated that this might be the trigger for postpartum depression because progesterone levels build up during pregnancy. They become quite high during the third trimester of pregnancy. The progesterone and this metabolite allopregnanolone were made by the uterus. By the placenta, and to maintain uterine function, et cetera. And that when the baby was born at parturition, the levels of both progesterone and allopregnanolone plummet to nearly zero. So they go from very high to very low. During the course of pregnancy, these receptors downregulate so that people who are pregnant, women who are pregnant, don't get completely sedated by this augmentation of this inhibitory transmitter. So to make a long story short, it was postulated with some good clinical data that was the trigger potentially for postpartum depression in susceptible women who had a predisposition to develop postpartum depression. So we did some early studies at Sage where we simply gave this compound, allopregnanolone, back to women who had postpartum depression, initially via an IV infusion. We gave it back to them for about 60 hours or so, and then we tapered it down. And so we allowed them to sort of withdraw from this naturally. And it turned out this was a very dramatic effect on treating the symptoms of postpartum depression. In about 70% of women, they responded almost immediately. Within 24 hours, they were not depressed anymore. It was really amazing. Didn't work in everybody, but it worked in the large percentage of women with postpartum depression. Now, it turned out that an IV formulation was not very compatible for commercialization purposes. You had to go into a clinic, you had to be watched, et cetera, et cetera. So the scientists at Sage came up with an oral form of this compound allopregnanolone, a synthetic derivative called zuranolone, which could be taken orally, and it has been proven and now is approved by the FDA for treating postpartum depression. A very remarkable, really rational example of rational drug discovery.
I also think of it as an example of precision psychiatry, which is just sort of a nascent concept right now. But the biology for that particular patient population was, very direct and obvious to understand. But then, like, as so often happens in drug development, right, Biogen wanted to start in this small kind of well-defined population and then see if it could work in a broader population. And that ultimately, that didn't quite translate. What can be learned from that?
So that's exactly correct. We attempted at Sage in collaboration with Biogen to develop zuranolone, this drug that worked pretty well for postpartum depression. By the way, it was a 2-week treatment in women who had postpartum, and then we stopped, and they remained, for the most part, without depression. We call it euthymic. Subsequent, obviously, if they get pregnant again, they're at higher risk, et cetera, down the road. But we tried the same 2-week protocol, zuranolone, in treating major depressive disorder, and unfortunately, we did not meet our primary endpoint. So the FDA did not approve zuranolone for major depressive disorder. I remind you, it was a 2-week treatment. We looked early to see if it had a rapid effect. And in fact, in all of those Sage trials, it seemed to separate from placebo at day 3. Very early on, we saw a nice antidepressant effect. But unfortunately, at 6 weeks, with only 2 weeks of treatment, there was not much of a drug placebo difference. And the FDA was very worried about what we call durability. And this drug did not seem to be durable in that condition. Now, I still believe, and many people still believe, that this is an active antidepressant, and that we had done a more traditional depression trial, which would be 6 weeks in duration, with various things that reduce the placebo response, which, by the way, haunts these trials. And there are many drugs, active drugs, as you know, that don't separate from placebo because the placebo response gets too great in these trials. And so we've come back at Seaport Therapeutics, a company I've recently co-founded with some colleagues from PureTech, Daphne Zohar, Michael Chen, and we've developed an oral formulation of the IV version of this neuroactive steroid, allopregnanolone. We call it Glyphallo, and we're now able to administer it as an oral drug. And so we've come back now to design a depression trial. This is in major depressive disorder, not postpartum depression. But in this case, we're going to treat for 6 weeks. We're not going to look too frequently at mood ratings because that enhances the placebo response. And we've also sort of focused in on a subtype of patient population, those that are depressed that have high levels of anxiety. We call it depression with anxious distress. And we have a theory, at least a hypothesis, that this drug, by virtue of its mechanism, is going to work better in that subgroup. So it gets back to your point about precision medicine, precision psychiatry, which, by the way, is a big challenge in psychiatry, but it's happening. And so that's the goal of us at Seaport, is to take a drug that we think might work because we have evidence in postpartum depression, reformulate it in a way that allows us to administer it orally well, and come back and test it to see if it does work in major depressive disorder.
Very good. Yeah, and so you mentioned Seaport's drug delivery methodology. So this is a new way to make small molecules, I guess, more orally bioavailable. So drug delivery is one of those things that have historically hindered psychiatry, one of many parameters, but certainly an important one. I don't know if you want to talk a little bit about that, but then just broaden it out to like other solutions to the drug delivery challenge that you're seeing.
You've said it really well. So sometimes you can take a drug that's got limitations. In this case, no oral bioavailability, doesn't get from the GI tract into the bloodstream very well, or it's chewed up too quickly by the liver, and you can reroute its absorption from going directly to the liver via the portal vessel through what we call the lymphatic system to get into the bloodstream. So drug gets to brain before it gets to liver. And we have a proprietary platform called Glyph that allows us to make a prodrug so that we can deliver the drug, but it fools the body in thinking it's a fat, a lipid, and it goes through the lymphatic system. And so beautifully, we can now take a whole variety of drugs. We've done this with 2 or 3 others now that, will never become drugs unless we can get them delivered properly, that now become good drugs. And so now we're back testing them to see if, in fact, we can demonstrate efficacy and safety in more traditional types of trials. So you're exactly right. And that's another way of really finding new agents that were really old agents that showed very good effects, but we'll never get to patients without this new technology.
I mean, it's a way to take this long history of clinical trial failure and use it for success, right?
Exactly right. Exactly right.
So, but in the field of drug development, there's lots of other things going on that are really interesting right now. I mean, obviously, Voyager has its gene therapy platform, and then there's this whole slate of companies using transferrin or other receptors in the blood vessels in the brain to ferry biologics across. Like, what are you keeping your eye on there?
I think that's very exciting. I think both the ability to find little viral vectors that get readily across the blood-brain barrier, which has been the limitation of what we call traditional gene therapy, and that's working out very well. A company I was involved in co-founding, Voyager Therapeutics, has come up with some very exciting viral capsids that allow us to deliver genes or silence genes in the brain where we can administer very low doses of the virus, which allows for the safety margins that you need to actually make this work clinically. And so they're off, doing some very exciting work, mainly in the neurological disorders, Alzheimer's disease, Huntington's disease, et cetera. So that's very exciting. And then you alluded to another very good one where it's called the Trojan horse approach, where you use the transferrin receptor or one of the receptors on the blood-brain barrier to allow us to get proteins directly, like antibodies, directly into the brain. Antibodies are too big to get in by themselves. So by engineering them to bind to this transferrin receptor, we can get the antibody into brain at levels that are much higher than would traditionally been possible by just injecting them IV. And so a number of the anti-amyloid antibodies are being used in such a way. And I think you're going to see much better clinical effects at much lower doses. So there's lots of advantages there as well.
Okay. Well, we only have a few minutes left. So let's turn to a hot topic of our time in psychiatry, which is psychedelics. The first one of these to go to FDA did not get approval, but the next ones coming through have kind of changed their development strategies a little. And I think there's a lot of optimism. One will come through, but there's also a lot of drawbacks to them in terms of how you have to deal with expectation bias, functional blinding in trials. And then even if they get through the market, can they really scale given that you have to monitor patients in clinic and, this and that? So just what's your take on the process?
So the initial observations that that psychedelic drugs like LSD have antidepressant and anti-anxiety, anxiolytic effects was made, again, somewhat serendipitously. This was work done by the late Roland Griffiths at Johns Hopkins. He was treating terminal cancer patients who had lots of depression and anxiety with LSD. And he found that, in fact, these patients benefited. They had, elevated mood, reduced anxiety, et cetera. And again, was this a placebo response because they were having a psychedelic trip? It was very an unusual protocol, as you can imagine. Um, and that led to more work by many, many people with both LSD and psilocybin to see whether or not these psychedelic drugs do have mood elevating effects. And the short answer is they do appear to have fairly dramatic and durable antidepressant effects, which frankly surprised me. And I've been doing this for a long time. There are still some major unknown questions. How big of an effect would this be? Because it's very hard to placebo control a psychedelic. Anybody who gets a psychedelic knows they're having a trip and that they didn't get placebo. So that expectation bias increases what we call the effect size. And so we're not quite sure, how dramatic the effect really is. To your point, clinically, these psychedelic drugs have to be administered in a very sheltered care environment with lots of people. By the way, they're administered very infrequently, maybe once, maybe a couple of times, but, and so, my prediction is that we will see probably the first approved psychedelic drug for depression here in the next year and a half, maybe 2 years at the outset. Now, the other cool thing is we believe we know how these drugs work. They work primarily by stimulating one of the 15 known serotonin receptors, the 5-HT2A receptor. And it turns out, Selena, that we have drugs that stimulate that receptor and produce a trip, and we have drugs that stimulate that receptor that don't produce a trip. And we've just recently figured out how that can occur. So there is a theory out there. We at Seaport share this theory because we have a compound called 2-bromo-LSD. It's a bromine on the second position of LSD that has been in humans already and has been shown not to produce a trip. Yet in these preclinical animal models, as limited as they are, this drug and others that stimulate this receptor that don't produce a trip seem to be active. So the big question on the table is, can you stimulate this 5-HT2A receptor and not produce a trip, a psychedelic experience, but have that robust antidepressant effect? And this is still to be determined. But to me, it's a very exciting possibility because then you could deliver these drugs in a more traditional way. You could write a prescription, patients could go home, they could take them, and it would be wonderful. So stay tuned. I'd say this is going to be in the next year to 2 years at most. We will know whether you can stimulate this receptor without getting a trip, a psychedelic experience, and still get robust antidepressant effects. And that, in my view, will be a very exciting thing for the field and for patients, still many of whom don't respond adequately to the current set of drugs that we have available.
Yeah, it would actually allow you to test with knowing what without the functional unblinding issues too. So
exactly.
You know the effect size.
Absolutely.
Yeah, all of those things. So do you think it's I don't know, because that when you don't get the psychedelic trip, but you're still stimulating the 5-HT2A receptor, it's through the way you're biasing the signaling, the G-coupled signaling?
That's right. You're up on the literature. The recent data suggests that it's the bias signaling that determines whether a drug that stimulates receptors produces the trip through different G protein coupling versus not producing the trip. And that's some recent data. And to me, that's very exciting because it gives us a molecular explanation for how this might be working. Now, the piece of the puzzle that we don't know yet is, do these non-psychedelic, we call them, by the way, neuroplastogens now because they enhance synaptic plasticity, do they actually have rapid antidepressant effects that are durable? And that's the unknown question, or the big question we've got to answer. And I'm excited to say that Seaport will be a part of that with our 2-bromo-LSD program. And there are others out there doing very similar work.
Yeah, that's very cool. Although the durability almost becomes less important because it's easier to
That's
redose in that
exactly right.
paradigm,
You could
right?
you could
Yeah,
administer this every
yeah,
day if you wanted to, or once a
yeah,
week, or twice
right.
a month. So
Yeah.
I'm really
Wow.
excited. I've my early career, I treated a lot of depressed patients as a private
Mm.
practitioner, and boy, I would love to have a drug that worked rapidly and effectively to treat mood disorders. That would be a great thing.
Okay, well, I'm afraid that's all we have time for today, but I think that's a great place to end it. It's very hopeful. And so I wish you all the best in those efforts, and thank you so much for sharing
Thank
some
you,
time with
Selena.
us. All
Much
right.
appreciated. Thank you.
Bye.
Brought to you by the 5th East-West Biopharma Summit in South Korea. In March, explore how to globalize Asia innovation and connect with key players across Asia's innovation arc, from India to China to Japan.
Kendall Square Orchestra provides the music for the BioCentury show. The group connects science and technology professionals and other members of the greater Boston community to collaborate, innovate, and inspire through music while supporting causes related to healthcare and education.